All right, this is the Jewish Exponent podcast. We are here with Dr. Elliot Barnathan, who is a Jewish Philadelphian. He is the husband of a rabbi, Shelley Barnathan, at Or Zarua on the main line. And he also made a significant contribution to Nobel Prize winning Dr. Katalin Karikó's career. And she, of course, won the Nobel for her work on COVID vaccines. We're going to go into that. But Dr. Barnathan, thanks for being here.
Thanks very much. It's great to be here, and hope to have an illuminating conversation.
Yeah, so you are an executive director at Johnson & Johnson. You have been in the medical field for decades at this point. You spent 10 years at Penn from 1987 to 1997. And it was while you were there that you met Dr. Karikó. And Dr. Karikó, of course, won the Nobel Prize in Medicine on October 2nd with Dr. Drew Weissman, who the Exponent covered because he is Jewish. And what they did, as Penn Medicine's website describes it, is invent the modified mRNA technology used in Pfizer BioNTech and Moderna's vaccines to prevent COVID-19 infection. Who knows how many deaths they helped prevent with their contributions. That is why they won the prize. But Dr. Karikó has had a more difficult road than Dr. Weissman for various reasons. And it was hard for her to get an opportunity early on, but you saw something in her, you brought her on, and you helped her career get started at Penn, which, of course, very important institution for anyone who wants to get started. So tell us about that.
Sure. So I actually spent a total of 24 years at Penn, having done all my undergraduate and medical training there as well. But when I started on the faculty, I was looking to recruit somebody into the laboratory who was really an expert in molecular biology.
Yeah.
And I was a cardiologist, and so I advertised for it, and Kate Karikó applied for the position. And so I hired her into my laboratory, and she had this idea that we could use RNA instead of DNA to do a gene therapy type of approach. Back at that time, it was completely heretical. People thought we were crazy.
Let me just stop you for 1 second.
Yeah.
Did she present that idea when you were looking to hire her? She had that coming in?
She did.
Wow. Okay.
She was interested in RNA right from in the early parts of her career.
Yeah.
And we had a variety of things going on in the laboratory at the time, but she was insistent that the better way to approach the problem of getting cells to make new proteins was using RNA. Mm-hmm. And she had a lot of ideas on how to stabilize the RNA, because RNA is very ephemeral. The body would degrade it immediately.
Right.
But she was able to, over the years, one by one, do incremental steps to try to make it a more and more viable therapy. And that's what we worked on for 8 years while she was part of my laboratory.
So you say it was heretical, that idea at the time.
Yeah.
You are an established person in this industry, and you see someone with a heretical idea and you hire them. So what went into that decision?
Well, there were a variety of things. First of all, she's a brilliant scientist, and she reads voraciously. And she was also the best scientist at the bench that I've ever seen. So she had a combination of skills. Plus, she was from Hungary. She was an immigrant. She came with virtually no money.
Yeah.
And she was a woman and a PhD in a department of medicine that, was, I guess, considered second-class citizen, if you will.
Yeah.
And but it was very clear when I first met her that she could make a tremendous contribution to the laboratory. And she ended up training a lot of scientists and students that came through the laboratory, and really propelled the whole field of mRNA therapy while we were working together in the laboratory.
What was it about her that jumped out to you when you were going through the interview process? So you learned everything you just described, as you worked with her, but what was it about her that jumped out when you first met her?
Um, she's really bright. She's very forward. I used to say working with Kate is like not drinking from one fire hose. It's like drinking from three fire hoses simultaneously. And she's very direct. So she comes forward right at you. And in fact, there's a funny story that she tells where I didn't actually, I wanted to keep interviewing. And she called up my secretary and she said, please have him look at my CV again. And so I said, okay, I'll interview you, but please bring your notebook. And so I looked at her notebook and I could see how carefully, meticulously she designed experiments. And I said, I want this person in my laboratory.
Almost like a beautiful mind type of thing. In
Absolutely.
there. Yeah.
Absolutely.
Yeah. So did you obviously liked her. Did you like her idea with mRNA as much as her?
You know, I understood what the problems were,
Yeah.
but I also saw somebody who was so passionately driven by the idea that I provided the space to allow her ideas to be fertilized and to grow.
Yeah.
And that's really now in retrospect, it was great that I did that. But back at the time, I was learning as much from her as from anybody. And there was challenging along the way that wasn't so straightforward.
Hmm.
But we made incremental progress, year after year. And then ultimately she made the key discovery, which was how to replace one of the base pairs of the mRNA. And that patent that she then filed for that is the basis for all of the COVID vaccines.
Yeah. So when she first started working for you, a lot of those other qualities that you mentioned, how good she was at the bench and things like that, started to jump out, right?
Yeah, I mean, the most interesting thing was she would read every science, Nature, all of the sort of highest level scientific publications. And she would
Yeah.
come in and talk about what somebody had found in a very obscure type of experiment. But
Yeah.
she pulled that information in and was able to apply it to what we were doing.
Yeah.
And that was just mind blowing.
Yeah. What are some other things she did like that also kind of showed you who she was even then?
I mean, she was meticulous in the design of her experiments.
Yeah.
And so here's an example. She was doing experiments, and when she would put the RNA into the cells, it caused an inflammatory response. And that was not good when you would treat animals with the mRNA.
Yeah.
And what she did was a control with a different type of RNA. It's
Hmm.
called transfer RNA.
Hmm.
And when she did that, the cells didn't have inflammation.
Hmm.
And what she then did is look at how the transfer RNA is built. And she found out that instead of the base pair of uridine, it had pseudouridine. And she said, let me take pseudouridine and put it in the mRNA
Hmm.
and see if the same thing happens. And in point of fact, it eliminated the inflammation and the expression went up by tenfold.
Hmm.
But it was her attention to detail in designing the experiment that enabled her to make that fundamental leap.
Hmm. Okay, so I want to quote now from a New York Times article about her that also goes into her experience in your lab. So it says, she and Dr. Barnathan planned to insert mRNA into cells, inducing them to make new proteins. In one of the first experiments, they hoped to use the strategy to instruct cells to make a protein called the urokinase receptor. If the experiment worked, they would detect the new protein with a radioactive molecule that would be drawn to the receptor. Most people laughed at us, Dr. Barnathan said. One fateful day, the two scientists hovered over a dot matrix printer in a narrow room at the end of a long hall. A gamma counter needed to track the radioactive molecule was attached to a printer. It began to spew data. their detector had found new proteins produced by cells that were never supposed to make them, suggesting that mRNA could be used to direct any cell to make any protein at will. I felt like a god, Dr. Karikó recalled. Do you remember this moment?
Absolutely.
Yeah.
Yeah, for
Take me
sure.
through it from your perspective.
Yeah, so what you do when you create an mRNA is you're essentially building base pairs that will instruct the cell to make a protein.
Yeah.
And the cell reads that, and when it reads that, if it reads it successfully, it then folds into the protein.
Mm-hmm.
And so the way we could tell whether these cells were making the protein was a receptor that sat on the surface of the cell. And so we took the radioactive molecule that would bind to that receptor and added it to the cells. And then you wash the cells away. And so if the receptor was really there, we could tell it with that radioactive. And so, we took the cells at the end of the experiment and we would put them in the gamma counter. And, as soon as you could see, if it's low like this, there's no binding. But all of a sudden, if it goes, way high, the experiment worked. And, and Kate was there with our chief laboratory technician, Alice Kuo, and the three of us just were ecstatic. Because that essentially said we can do what we want to do, and the rest is sort of figuring out which cell and how high that expression will be, et cetera.
So you had a sense at the time that you had made a profound discovery?
Well, I'm not sure about how profound it was, but
Yeah.
it essentially said we can use mRNA as a therapy,
Right.
and that all the doubters were wrong, and we can put it in a capsule, add it to the cell, and it can actually do what we said it would do. And that was fundamental. I mean, really, for sure.
Did you feel a little crazy before that when you were trying to accomplish something that no one said you could do? You and Kate?
I always believed in Kate because she was so smart
Yeah.
and so thoughtful, and also humble and very self-critical.
Yeah.
So if the experiment didn't work, she spent hours and hours trying to figure out why.
Mm-hmm.
But she always came back positive the next day with the next series of experiments.
Yeah.
And that type of enthusiasm was infectious.
Mm-hmm.
And so everybody in the laboratory was just really excited about how we can create a totally new type of therapy. And getting on board with that was just, it was challenging, and we all realized it was challenging,
Mm-hmm.
but we thought it was worth it.
She really does seem like the ideal of a scientist. Is that a fair way to describe it?
Absolutely. I mean, no question about it. And, and she loves science.
Yeah.
It was her drive, because you have to realize she was fighting being an immigrant, being a woman, being a PhD, and in an institution that, wouldn't agree to allow her on a tenure track, then not allow her to get promoted even in a non-tenure track position, then ultimately deciding, that they didn't want to continue having her employed there.
Mm-hmm.
It's really hard, but her interest was always driven by wanting to create therapies for people. And even when the vaccine finally, was created, it was only when it was actually given to people and that it was saving lives that she felt some sense of accomplishment, which
Yeah.
is amazing given how much work she had been doing for years and years before that with almost no recognition whatsoever.
All of these politics, I guess you would call it, that hindered her career in a way are talked about when she is talked about now with the Nobel Prize. But based on her quotes, based on what you're saying here, based on her focus in the lab, none of it seems to bother her really. She just wants to have a way to do the science. Is that just the outside perception, or is that really the case?
Yeah, that's sort of the outside perception.
Okay. Yeah.
That's because she's gracious and
Yeah.
she's humble,
Yeah.
and she's not interested in crying over spilt milk and
Yeah.
whatnot. But a lot of it was hurtful, very much so. And
Yeah.
she wrote a book recently, which just came out.
Okay.
And if you want to read the real story behind everything, she doesn't hold anything back in the book.
Yeah.
But it was all challenging and difficult for her and whatnot. But she also, despite all of that, it was her interest in science, her love of science, and her interest in the next experiment and getting the answer to the next experiment that
Mhm.
kept her going.
Right.
And when she talks about it, it's her love of science that enabled her to not be so obsessed with the career or the title or the money or any of the things that were deprived from her,
Yeah.
that she was able to keep going.
She could really lose herself in it, and that would help
Yeah,
her.
exactly.
Yeah.
And that was her ultimate reward.
Mhm.
Now, at the end of the day, you could say, well, isn't the ultimate reward winning the Nobel Prize? But she would say, no, it's
Yeah.
not winning all the prizes. It's the fact that she was able to use her scientific knowledge to create something that ultimately helped people. That's her main reward.
Yeah. So I want to go back to a couple more passages from this article for a second. Uh, After you made that discovery, she and Dr. Barnathan were on fire with ideas. Maybe they could use mRNA to improve blood vessels for heart bypass surgery. Perhaps they could even use the procedure to extend the lifespan of human cells. Um, how did you guys feel about the future in that moment?
You know, for us, it was the difference for mRNA and DNA is DNA could change the cell forever.
Mhm.
Now, that may be good or that may be bad. If you're a diabetic and you need insulin, well, then you need insulin all the time. And so, or you're a hemophiliac and you don't have the gene that you need to have blood clotting, you need that all the time. But
Mhm.
what we needed to do was find a therapeutic approach where you only needed the protein for a short period of time, and then it would go away and it would have no chance of causing cancer or problems with the cell that could damage the cell. And so it created a need to think differently about how this therapy would work. And so that's why the blood vessel, for example, we only wanted it to express the protein for a short period of time until the cells recovered and then could start making the proteins on their own.
So you are going to accompany Kate to the Nobel Prize ceremony
Yeah,
in Stockholm in December. As the article that I'm quoting from says, you left the university shortly thereafter. You accepted a position at a biotech firm. Kate went on with her career, but you guys obviously maintained a relationship through all those years and decades.
absolutely.
Yeah.
We've always maintained, kept in touch. And I still remember when she invited me to come back to Penn to show me the exciting experiment with animals where she injected hemoglobin, the mRNA for hemoglobin, or for the protein that makes the cells make hemoglobin, so erythropoietin,
Mhm.
and showed that the hemoglobin went sky high,
Mhm.
and that was the sort of proof in animals
Yeah.
that mRNA therapy would work.
Mhm.
So we've stayed in close touch over the years, and she's just a great person, a dear friend. And with all of the awards, she's been kind enough to invite me to some of the award ceremonies and whatnot.
Mhm.
And both myself and David Langer, who was a student in my laboratory, ultimately became a neurosurgeon. And when I left Penn, David was kind enough to sort of get the neurosurgery department to have a home for Kate for the next phase at
Yeah.
Penn, and he'll be coming as well. So we're really excited to be at her side and just be appreciative of the final and large recognition of an entire body of work over many years.
Yeah. So I think we all need help in life. No one does it alone. You helped her and she went on to accomplish this great thing. She is acknowledging that by inviting you to this ceremony. What does that mean to you?
I'm truly humbled. It's, as scientists and as physicians, you always talk about going to Stockholm as something that you would never have happen in
Yeah.
your life. And the fact that I can go to Stockholm and celebrate one of the people who I helped in a small way,
Yeah.
that's the ultimate, really. For somebody who cherishes teaching and mentoring as I do, I've always said that my greatest reward in life is not by what I accomplish, but what my students accomplish and the colleagues. And so to me, it's going to be just an incredible honor.
Dr. Barnathan, thanks for joining us.
Thanks very much. Great to be here with you.